Hello, I'm Rob Noble. I'm a lecturer at City University of London. Prior to that, I was a postdoc at ETH Zurich and the University of Zurich in Switzerland, where I did most of the work I'm going to present about when, why, and how tumor clonal diversity predicts survival. This work was inspired by clinical studies such as this one by the TracerX Renal Consortium. In this study, they found that in kidney cancers, patients whose tumors have higher intratumor heterogeneity in terms of the genetics of those tumors tend to have shorter survival times. That's shown in the top left-hand panel here. They also found that tumors with high genomic instability, abbreviated here as GI, also tended to confer lower survival times. And when they looked at both these factors together, as in the top right, they found that particularly in tumors that have low genomic instability, high intratumor heterogeneity is a predictor of worse survival. However, when they adjusted their analysis for stage and grade, the conventional biomarkers, they found that these differences were no longer significant in their cohort, although they did find they were significant in the larger Cancer Genome Atlas cohort shown here in the bottom row. What I wanted to understand was why these patterns emerge and how robust they are. So I developed a model of tumor evolution. This is a spatial stochastic model of tumor evolution. And within this model, what I looked at is if we observe simulated tumors at a certain size, shown here is the example of 250,000 cells, that's the measurement size, and then we measure how long it takes these tumors to reach some larger size, in this example 1,000,000 cells, we see whether that waiting time for the subsequent growth correlates with predictor variables that we measure at the earlier time point. So in this case, we measure the mean cell division rate of the cells within the tumor at the early time point, and we see whether that correlates with the future growth rate of the tumor. What we find is that if we look at clonal diversity, we can find either negative or positive correlations. This is somewhat unexpected. So earlier you saw positive correlations in the TracerX renal dataset. Here we find either positive or negative correlations. Particularly, we see negative correlations when the tumor is observed at a relatively large size, as shown here with those purple dots. When we investigate within one of these cohorts, we see the underlying reason for this negative correlation. So here you see a negative correlation between clonal diversity and mean cell division rate, which carries over to a negative correlation with the tumor growth rate. If we examine a tumor that has low clonal diversity but high cell division rate, we see that such tumors have undergone a succession of selective sweeps. In the diagrams here, each clone, each combination of driver mutations is shown as a different color, and in the bottom row, the clones are colored according to their fitness, the mean cell division rate within that clone. And you can see that beneficial mutations, driver mutations, have swept through the population, which increases the growth rate of the tumor. Conversely, if we look at a tumor that has high clonal diversity and low cell division rate at the final time point, we can see that the reason for this is that in these tumors, multiple clones have emerged with similar fitness, and these have impeded each other's growth. This is known as clonal interference, and it slows the spread of beneficial mutations, thus slowing the growth of the tumor. If we look at many cohorts of tumors, we find that those that have higher clonal turnover tend to have negative correlations between clonal diversity and average growth rate, whereas those that have very low clonal turnover tend to have positive correlations. But in reality, of course, even tumors of the same size and type vary in their underlying biological parameters, and particularly in genomic instability and driver mutation rates. So if we instead look at cohorts of tumors in which each tumor is initially assigned a randomly chosen driver mutation rate, and these driver mutation rates vary within the cohort, we find consistently positive correlations between clonal diversity and future growth rate. And this is largely because clonal diversity co-varies with this underlying driver mutation rate. And that correlation is robust to biopsy sampling error. So here we mimicked the multi-regions biopsy sequencing method that was used in TracerX renal and other studies, and we found that the correlations are robust to this sampling method. Finally, what we looked at was if we grow tumors and we allow them to acquire rare resistance mutations, and then we subject these tumors to treatment when they reach an endpoint size, in this case 1 million cells, then any surviving cells are allowed to regrow, and we measure how long it takes the tumor to rebound from its pre-treatment time point to reattain its pre-treatment size. This is the progression-free survival time. And we see whether that progression-free survival correlates with observations we make of the tumor just before treatment. So in this case, we've observed clonal diversity just before treatment, and we find a correlation there between clonal diversity and survival, just as in the TracerX renal study. And here you see the results presented as Kaplan-Meier curves in the same format as TracerX. So the top row is the model results, and the bottom row is TracerX. However, if we adjust our results for the underlying driver mutation rate, we find that the correlation with diversity is no longer significant. So what we conclude is that diversity is only useful in as much as it's a useful proxy for underlying biological parameters that we can't measure. So I'd just like to thank Cécile Lesseur and Jean Burley, who were instrumental as master students in this work, and Michael Hochberg, who was my supervisor when I started this work in Montpellier. Thank you very much.